So hi students, this is our last portion for chapter 6, and in this part we will be talking about the peripheral nervous system, including the autonomic, and we'll also talk about what protects the brain. So the peripheral nervous system transmits signals between the CNS and between the effectors, and remember the effectors can be skeletal muscles on one side, and that would be your somatic, or on the other hand could be cardiac muscles, smooth muscles, and glands, and that would be the autonomic nervous system. The peripheral nervous system is made out of 43 pairs of nerves, so 43 on the right and 43 on the left. And we have 12 pairs coming out of the brain. We call those 12 cranial nerves, plus the 31 pairs of spinal nerves coming out of the spine. You add those up, those will make up the 43 pairs of nerves. The 31 pairs of spinal nerves are named according to what part of the spinal cord do they come out of. So I'll show you a picture to illustrate that. So the nerves coming out of the cervical spine are known as the cervical nerves, and we have 8 of those. The nerves coming out of the thoracic spine are 12. We call those thoracic nerves. The nerves that come out of the spinal cord in the area of the lumbar region are known as your lumbar nerves, and those are from L1 to L5, so there are 5 of them. The sacral nerves come out of the sacral area, and those are also 5. And then we've got one coccygeal nerve, and we symbolize that with the CO. Okay, to kind of distinguish it from the C1 through C8 that symbolizes the cervical nerves. The CO is a symbol for the coccygeal nerve. Now the 8 pairs of cervical nerves, those innervate skin and muscle of the neck, shoulders, and upper limb. While the 12 pairs of thoracic nerves, those innervate the skin and muscle of the chest and the upper abdomen. The 5 pairs of lumbar nerves, those are associated with the innervation of the lower abdomen, the hips, and the lower limbs. 5 pairs of sacral nerves are associated with the innervation of the genital area and the lower part of the digestive system. While that one single pair of coccygeal nerve is responsible for innervating the skin in the area of the tailbone. So again, when you add all of those up, you get 31 spinal nerves pairs. So 31 on the right, 31 on the left. And we already went through the breakdown of these nerves. Now the peripheral nervous system, we can break it further down into a sensory afferent division or a motor efferent division. Our spinal nerves are mixed, meaning that they carry both afferent and efferent fibers. While that is not the case when it comes to the cranial nerves. Some cranial nerves are just purely sensory, while others can be purely motor, and so on. But the spinal nerves are mixed. And we already talked about how we have the ventral and the dorsal root, where they combine together to make the mixed spinal nerve. Okay, the sensory, the motor division, again, takes information away from the CNS or away from the spine and would down to the effector. The effector, again, could either be a muscle or a gland. And that further divides the efferent neurons into somatic, that would be to your skeletal muscles, so voluntary, and autonomic nervous system, and that would be to the cardiac muscles, smooth muscles, and glands, so the parts that you do not have any voluntary control over. Again, this is the division of the efferent part of the peripheral nervous system, so the motor part. Again, you have somatic and autonomic. So remember, you have neurons that are going to go from the CNS towards the effector. In the case of your somatic system, it is one single neuron that goes all the way from the CNS all the way to the skeletal muscle. So the cell body would be in the brain, and the axon will go all the way down until it reaches the skeletal muscle. So you can imagine it is one long axon. So it is a single neuron. It innervates the skeletal muscle cells, and when an action potential reaches the muscle, it leads to muscle cell excitation and muscle contraction. The autonomic nervous system is different. Instead of having one single neuron, it's actually made out of two neurons, and these two neurons connect at the synapse. So there's a presynaptic neuron and a postsynaptic neuron. These are going to innervate smooth and cardiac muscles and glands, and they can either excite the muscles or inhibit the muscles. Okay, so in your case of somatic muscles, there is no such thing as inhibiting a muscle. Okay, you just don't excite it. But in the autonomic nervous system, we can excite or inhibit the effector. So you can see here in the case of the somatic nervous system, there is one neuron where the cell body is in the CNS. The axon goes all the way to the skeletal muscle or to the effector. Again, that is the case in the somatic nervous system where you have voluntary control over your skeletal muscles. In the autonomic nervous system, where your effectors are involuntary things like smooth muscles, cardiac muscles, or glands, that is made out of two neurons, and they meet each other at the ganglion. So we have a preganglionic neuron and a postganglionic neuron. The anatomical and physiological differences lead to the further subdivision of the autonomic nervous system into sympathetic and parasympathetic. The origin of the fibers, whether sympathetic or parasympathetic, vary. So the origin, when it comes to the parasympathetic fibers, they start either from the cranium in the brainstem or from the sacral part of the spinal cord. So we call the parasympathetic fibers, their origin would be craniosacral. So it's kind of like you have the spinal cord in parentheses, parasympathetic parentheses, so craniosacral origin. And again, we're talking about the origins of the preganglionic fibers. So these neurons, the preganglionic neurons, in the case of the parasympathetic nervous system, would be originating either from the cranium, brainstem to be particular, or the sacral area of the spinal cord. While the preganglionic neurons of the sympathetic fibers originate from the thoracic area and the lumbar area. So collectively, we call it the thoracolumbar origin. The GI tract actually has its own nervous system known as the enteric nervous system. And remember when we were kind of talking about the different neurotransmitters, we said that most of serotonin is actually found in the GI tract. And this is why, because the GI tract has its own, pretty much its own built-in nervous system. Okay, and we could also classify that into sensory and motor and inner neurons. So the enteric nervous system is kind of like a built-in separate nervous system. Here you can take a look at, on this side, these are the origins of the parasympathetic fibers, and on this side are the origins of the sympathetic fibers. These are the organs, these are the different effectors. So if you look at the parasympathetic area, you see again that the preganglionic fibers originate from the craniosacral area. So again, kind of parentheses. Cranium, to be very specific, it would be from the brainstem or the sacral spine. These fibers are very long. The preganglionic fibers are very long. The ganglia can be very close to the effector, even sometimes right on top of the effector. Okay, and they have very short postganglionic fibers. In the case of the sympathetic nervous system, the preganglionic fibers originate from the thoracolumbar area, so the thoracic and the lumbar part. They have very short preganglionic fibers. You can see the ganglia right here, while the postganglionic fibers are much longer. So again, just to keep, to repeat everything all over again, looking at the paragraph as opposed to the image, the sympathetic division is also known as the thoracolumbar division. It has a short preganglionic fiber and a long postganglionic fiber. We'll talk about the neurotransmitters when we get to another slide. Okay, this is your fight or flight response system. The parasympathetic system originates from the craniosacral area. These have very long preganglionic fibers and very short postganglionic fibers. And again, we'll talk about the neurotransmitters in a bit. The parasympathetic system is also known as the rest and digest system. So this is the part of the nervous system that takes over when you're about to fall asleep, when you are sleeping, and after a meal, when you're digesting a meal. Now in this image, you can see the neurons and the different neurotransmitters with the receptors. So the one on top represents the somatic nervous system. You see here again, the cell body originates from the CNS, one long neuron until it reaches the effector. The neurotransmitter at the neuromuscular junction is acetylcholine, and the receptors on the skeletal muscles are nicotinic cholinergic receptors. In the case of the autonomic nervous system, and that's the lower portion of this image, we break it down into parasympathetic, where the preganglionic fiber originates from the craniosacral area. It has a long preganglionic fiber, and then it synapses with a very short postganglionic fiber. The neurotransmitter at the ganglion is acetylcholine, and the receptors here are nicotinic cholinergic as well. Now this postganglionic fiber will reach the effector, and the effector in the case of the parasympathetic nervous system would have muscarinic cholinergic receptors. So you see here that both in the case of the parasympathetic division, both the pre and the postganglionic fibers release acetylcholine. The receptors in the ganglion are nicotinic, while the receptors on the effector are muscarinic. Now down to the sympathetic portion. Sympathetic, the preganglionic fibers are short. They originate from the thoracolumbar area, and then they synapse with a very long postganglionic fiber until it reaches the effectors. In the area of the ganglion, it is also acetylcholine as your neurotransmitter with the nicotinic cholinergic receptors. The postganglionic fiber, though, releases norepinephrine, and hence we have the adrenergic receptors on the effectors. The ganglia that reach the top of the kidney are a little bit different. Those are not going to produce a postganglionic fiber. These ganglia are going to change and collect to make the adrenal medulla. The adrenal medulla is going to release epinephrine. Epinephrine now is going to be considered a hormone because it will go through the bloodstream to reach the effector. The receptors on the effector are also adrenergic receptors. So to kind of summarize the different kinds of neurotransmitters, in the somatic, they're acetylcholine. In the ganglia, all of the ganglia are acetylcholine as well. All of these have nicotinic cholinergic receptors, whether it's in the somatic system or in these ganglia right here. Parasympathetic, postganglionic fibers release acetylcholine, but the receptors are muscarinic on the effector. In the case of the postganglionic sympathetic, those release norepinephrine with adrenergic receptors on the effectors. So we are going to be repeating a lot of this all over again, okay? So in addition to all of these neurotransmitters that we described, the postganglionic neurons can recognize or recognized as there's a widespread of postganglionic neurons that do not recognize either adrenergic or cholinergic receptors, and we are going to ignore that fact because that is just a little bit too much detail than what we would need to know. Okay, some neurons are going to use nitric oxide, and again, we're kind of, put that to the side for now. We talked about nitric oxide when we talked about neurotransmitters, so I don't want to really bring it up and mess up this whole autonomic nervous system. The vast majority of the cholinergic receptors in the autonomic nervous system are of the nicotinic kind, okay, except in the postganglionic parasympathetic. Those are muscarinic. So the acetylcholine receptors on the smooth muscle, cardiac muscle, and glands, which are your effectors, are muscarinic receptors. The cholinergic receptors, in the case of the skeletal muscle fibers, those are somatic motor neurons, and again, they release acetylcholine, and the skeletal muscle fibers have the nicotinic cholinergic receptors. The adrenal medulla is a very special case of ganglion, where the ganglia fails to produce postganglionic neurons, but rather the adrenal medulla cells are going to produce epinephrine, release it into the bloodstream, and that's why we're going to call it a hormone. And when it reaches its effector, the effectors have the adrenergic receptors as well. Now, a lot of the effectors, when we're talking about the autonomic nervous system, have dual innervation. So they're innervated by both the para and sympathetic systems. They would have the opposite effect. So, for example, if the parasympathetic nervous system stimulates the digestive system, the sympathetic would inhibit the digestive system. Okay, so again, the organs would have dual innervation, but the effects would be the opposite. These four tables are not tables that you would have to memorize. They're up there for interest. Okay, so that you can see how the sympathetic fibers and the parasympathetic nervous system have opposite effects on the same organ. So for instance, the SA node of the heart, or the pacemaker of the heart, your sympathetic nervous system will stimulate the receptors, the beta 1 receptors, leading to an increase of heart rate, leading to tachycardia, while the parasympathetic nervous system decreases the heart rate. Okay, so again, you can kind of go over these tables on your own time and kind of take a look and compare and contrast the effect of the sympathetic and the parasympathetic on the different parts of the body. So what are the parts that, we kind of already talked about everything. We just want to talk about how do we protect these very important structures. So there's obviously the bone. We're talking about the brain now. So we've got the bone, the skull, and then we have three meningeal layers. So the first one, from superficial to deep, the first one is the dura mater. That is made out of tense connective tissue. And underneath it, there is the subdural space. The subdural space has venous blood that will be collected and, taken over to the heart. Underneath the dura mater is the second meningeal layer, known as the arachnoid. The arachnoid gets the name because it does really look like a spider web. The subarachnoid space, which is the space beneath the arachnoid matter is full of CSF. Now the third and the last layer is known as the pia matter, and the pia means the mother. Okay, so you will find that the pia matter is actually literally attached to and kind of like hugging the brain tissue proper. And it even will extend down into the sulcus and then reflect back up. And again, you literally have to peel it off of the brain in order to remove it. So it's a very thin layer. It kind of looks like saran wrap, if you know, if you've ever seen what that is. And again, you'd peel it off of the brain. But the dura matter is easy to separate. The arachnoid looks literally like spider web, so it's very easy to remove. It's the pia matter that sometimes is hard to remove. Okay, there are cavities within the structure of the brain. These cavities are full of cerebrospinal fluid that are made by the ependymal cells. So remember back way over at the very beginning of the chapter, we talked about the ependymal cells that line the cavities of the CNS. Well, those ependymal cells collect together or join together to make a plexus known as the choroid plexus. And that choroid plexus secretes the CSF. CSF then circulates within the ventricles. So we have these two lateral ventricles within the right and the left hemispheres. And then it would go to the third ventricle, which is found right in the middle between the two cerebral hemispheres. There's a duct known as the cerebral duct of Sylvius, or the duct of the midbrain, because it is right in the middle of the midbrain. And then that connects down to the fourth ventricle. Then the fourth ventricle, from there we have the central canal that goes all the way down through the spinal cord. It would leak out of these ventricles, okay, in order to surround the spinal cord and surround the brain. So I'm talking about the CSF, obviously, that is going to leak out. So we have the bones, whether it's the skull to protect the cranium or the backbones, the vertebral bones to protect the spinal cord. We also have the meninges. The dura is the most durable. It's the thickest of the three layers, and it's also the most superficial. The arachnoid matter is the middle layer. And then the pia is the thinnest layer, or the thin layer, and it is directly on top of the brain tissue. Also the spinal tissue. CSF, again, protects and cushions these structures. CSF is full of nutrients, so it's got glucose and different ions and so on, and this is how it helps in the nutrition of the CNS. Subarachnoid space, that is between the arachnoid matter, well, that's spelled wrong, and the pia. Okay, it's full of CSF, and the pia matter is just deeper to that, okay? And again, the pia matter is right in direct contact with the actual nervous tissue. So a little bit clinical on meningitis. So meningitis is the inflammation of the meninges, okay, that you could see the inflammation or the effect of the inflammation in the CSF. So if you want to diagnose meningitis, you can get a specimen of the CSF through a lumbar puncture, which would be inserting a needle in the lumbar area, aspirating a little bit of CSF, and you could take a look at it under a microscope, culture it, see if it's a virus or a bacteria or so. Well, culturing would be more for bacterial. You can kind of find what is the pathogen causing the infection and so on. CSF, we already said, is made by the ependymal cells. These ependymal cells are microscopic cells, so when they gather together, they could give you something that you could actually see by the naked eye. That is known as the choroid plexus. Choroid plexus will produce CSF. It produces at a pretty good high rate, where we are replenishing the amount, the totality of the CSF about 3 times a day. CSF will circulate through the ventricles down to the third ventricle, and then from the third to the fourth ventricle. The third and fourth ventricles are connected via the aqueduct of the midbrain, and then down through the spinal canal or the central canal and the spinal cord. As I said, the CSF will eventually flow out and surround the spinal cord and the brain. It'll circulate within the subarachnoid space. Hydrocephalus literally translates into water and head or brain. So that means water on the brain, where we have an accumulation of CSF in the brain. It could be caused by a tumor. For example, that tumor would be known as an ependymoma. So remember, the ependymal cells produce CSF. So if you have a tumor in those, we are producing too much CSF. Or it could be something obstructing the pathway of the drainage of CSF. Okay, these are severe. That is a more severe case. Okay, and that you'd have to correct it surgically, where you introduce a shunt, shunting the excess CSF from the cranial area. The shunt will go all the way down and drain the excess CSF into the peritoneal cavity, which is in the abdomen. As this kid grows, you'd have to replace the shunt with longer ones. Okay, because if you don't do that, and there is that accumulation of CSF leading to the increase of pressure inside of the brain, that could lead to the destruction of neurons and permanent brain damage. We already touched base a little bit on the blood-brain barrier, where we mentioned it is a barrier between the nervous tissue and the blood in the capillaries. It is needed to prevent certain things from reaching the neurons, and it is formed by, I'm actually going to go to the picture. So it is formed by if you guys remember, the astrocytes and these little feet. So these feet form part of that barrier. We also have the basement membrane of the capillary, and then these tight junctions between the endothelial cells of the capillary. So all three of these layers are part of your blood-brain barrier. So again, the tight junctions between the endothelial cells, the basement membrane of the capillary, and these feet or extensions of the astrocyte. Again, if you want to give medication that would have any kind of effect on the brain, that medication would have to be able to cross the blood-brain barrier in order to reach the neurons. Okay. Now, I believe this is the last slide to talk about, and that would be stroke. So unfortunately, stroke is a common thing that occurs where there is brain damage caused by a vascular disease. So it could either be bleeding, or it could be due to a blood clot. But again, any brain damage caused by a vascular disease is known as a stroke. And so although the brain is about 2% of our total body weight, but it takes about, from 10 to 15% of our total blood supply. The neurons are very active even when we're sleeping. Okay, so they need more blood than their weight actually would specify. Okay, when the neurons do not have enough oxygen or glucose, even if it's for a few minutes, that could lead to cellular death. And remember, we talked about how that could spread out to the healthy cells, the healthy surrounding cells. Now, this would be a very good time to celebrate the end of Chapter 6.